So I wanted to start off by just thinking about freshwater ecosystems and just more broadly and our biodiversity. So our freshwaters in general support, that's only 1% of our Earth's surface, and yet we have 1/3 of all the vertebrates found within our freshwaters. And worldwide, the freshwater biodiversity is among the most imperiled when you compare our three ecosystems that essentially the freshwater taxa, that includes our fishes, our amphibians, our turtles, our mussels, our macroinvertebrates, they're at greater peril than in our marine and terrestrial ecosystems. And a lot of that has to do with the connectivity, between habitats that the different species use, as well as the variety of endemic species or species that tend to be only found within certain areas. So in general, sampling for fishes and aquatic species encompasses electrofishing. That tends to be one of our main, traditional approaches that we use in our freshwater environments. People also use minnow trapping, snorkeling, but what essentially comes from that is it's based on visual detections and counting. And so you have to have that taxonomic expertise in the field to identify the different species that you're processing. And so it's dependent on those people, being part of your field crews to make those assessments. Often limited to assessments of adults. Think about, our salmonids. We have a couple different programs, that target juveniles, but most of the assessments are of, our adult fishes. And it's often of listed species. And so, for example, you think about, main stem Willamette River, assessments are coming through for Chinook and steelhead. And, what do we really know about, sand rollers? And what do we really know about three-spine sticklebacks and a variety of the species that are considered less charismatic? And those assessments often occur in summer with the traditional approaches because that's when it's safe to access our streams and rivers. And so that's when the primarily time of the year is that we have an understanding of what's going on. And so we don't actually know what's happening in some of our freshwater systems in fall, winter, and spring, when access to those locations is either not possible because of snow or it's not safe, to actually be in the streams, wading and doing some electrofishing. So just kind of a visual look at what that actually looks like in our system. So oftentimes you end up with, a focused area in main stem parts of the river, sometimes main stem parts of tributaries, and then focused only on those listed species. And you don't necessarily have a complete understanding of all the species there, or population assessments across many of them. And so I feel like that's a good way to move into and thinking about eDNA. eDNA is short for environmental DNA, and it refers to DNA that can be extracted from soil, air, or water. I'm going to be focusing on water today, but do keep in mind that you can actually do the same approaches I'm going to present today for soil samples. You can actually take, the same thing on trees. People are doing like paint roll brushers on trees and identifying different species using the trees. They're doing in old growth forests. They're taking scoops of soil and looking for red tree bulls. There's just so many different ways you can do the approaches I'm going to present and explain to you from water in the different environments that would be soil or air. And also realize that I'm going to present results that are focused primarily on fishes, amphibians, our macroinvertebrates, and some of our pathogens. But this is broader than that. You can do it on plants. You can actually identify riparian animals that are using stream corridors if you're targeting them. And so it's broader and it has the possibility to identify plants and animals and then across multiple taxa, all from the one sample. So again, just to kind of come back, so you can find the DNA of the different animals, plants, and organisms on the sample. So think about essentially what eDNA is you're taking a scoop of water from your stream or river, lake, wetland, and you're processing it through a filter. So what you're seeing there is an actual photograph of a filter. In that dark kind of browny yellow color is, the Willamette at that time of the year. And so the DNA sits on top of the filter. Then we bring it back in the lab. And it looks like, a chemistry experiment where you have 6 steps and you're adding, the little pieces, and essentially you're extracting the DNA off the top of the filter into a little vial. And I don't know if you've ever seen at like the museum, up in Portland, but if you hold DNA up to the light, it looks viscous. It looks chunky. It's clear, but if you hold it up to the light, you can actually see it in your vial and in a clear sample. So essentially that's what you're doing. You're taking the DNA from the environment, you're in the lab, you're taking a couple steps to get it to the point where at this point you would then pipe it out a set amount and you'd run it on the machine and you would identify what species are there. So I'm going to talk about some different approaches today where you can take one species off of that sample and some where you're trying to look for multiple species off of that sample, and talk about some pros and cons of the different approaches. So as I go through the different studies that I'm going to present here, it's going to be important to think about the study system you might be interested in studying. Are you interested in streams, main stem river, some wetlands? Just because there's different choices you might want to make as you're thinking about sampling those lakes, reservoirs. The focal species that you might be interested in, is it 1, is it 10 species, or is it 100? Because then there's different choices you would make then based on which species you might be focusing on, and what the objectives of the project are. Do you just want to know if the species are there? Do you want to know what their distribution is? Do you want to have a sense of relative abundance? What are essentially you trying to understand? And that'll also help you make some decisions along the way in which approach that you might use. So I'm first going to start off presenting what's called eDNA eDNA barcoding, or single species eDNA, sometimes it'll be called. This is the eDNA approaches that's typically used across most labs, especially in North America. I would say as a community of North American scientists, this is the approach that's primarily being used right now. You take a sample in the field. Now, the example I'm showing here is a sample with a bottle. You can also do inline filters. So you don't have to do it as I've shown here. There's a variety of ways to collect that water sample. Some people also collect from one location in your water body, and some will collect 3 and combine into one filter. So there's different things you want to be thinking about as you think about what you're doing. So you collect your water, you would filter it through your filter. And then with the single species approach, this is where it's going to diverge from the one where you're going to look for multiple species that I'll explain to you, but you're essentially going to only focus on one. So in my example here, we're interested in coastal cutthroat trout. So essentially you're ignoring all the DNA on the filter. It's there, but you're not asking it to tell you anything about if it's there or not. You're just asking for it to tell you about the DNA for cutthroat trout. So that's why I've X'd it all off there, to essentially just identify it's there if you would want to ask questions related to it, but in this case, you're not. With this approach, it's the most straightforward related to genetics. You're essentially just looking for so you'd have what's called a primer, and you set it up so that it will only detect coastal cutthroat trout. It's the most work with this approach. It's the most work in advance in the lab, because you have to make sure it's only going to detect coastal cutthroat trout and not a sister species like rainbow trout. Same thing for bull trout. You want to make sure it's only going to detect bull trout and not brook trout or Dolly Varden. It's not really a thing here for your systems. But as you would take a primer out of the literature and potentially want to use it for your watershed, you want to ask the question, are there any sister species that haven't been tested against this one, as I'm going to pull this primer out and potentially ask this lab to use it for us. So you always want to check where that primer was developed, what species it was tested against, so that you won't get hits in your system for related species, whether they're, and in our case, non-native, or native related species, because if you're interested in bull trout, we do have brookies and a variety of the tributaries, so that would be something you just want to make sure. So essentially, that's the workflow for how single species eDNA works. So this is an example of the setup that I like to use in the field. Again, there is no right way to collect the samples in the field. The most important part is that how you collect it in the field needs to make sure it can be ran by the lab you're interested in running your samples by. So you want continuity between the filter, how you're preserving it in the field, and the lab that you decide to use. I think it's just important to note that there is no right way. Every lab does it a little different. Some want it folded into fourths, some want it wrapped like a flute, some want it preserved in lysis buffer, some want it preserved with, desiccant, some want it preserved in alcohol. It all works fine generally. I do have to say we've had some issues with alcohol. on gravel roads because it actually will eat the filter based on the chemistry of the alcohol mixing with the filter. If the filter is of a certain substance, that's cellulose acetate or cellulose nitrate. Obviously, it was developed in a place that had paved roads because in our roads here in Oregon, many of them are gravel. So those are just things to think about. But it's not that it's necessarily wrong to go that way. But when you get your sample in from the field, you're coming in with the filter. That's the most important part, is that with the lab you're going to, is that first step into the lab, the filter's prepped in a way that they'll take the filter in for their work. I like this setup because we collect with bottles in the field. You can also use whirl packs, but essentially it allows you to collect from multiple locations and put into one filter. So in our streams, we filter 3 liters of water, and I can collect from 3 different spots all within the same area and put it through the one filter. And then I can run 4 replicates at a site at one time. And so that just allows us to be more efficient in the field. Some people set up allow you to just process 1 replicate at a time. So you have to process one, maybe it's 20 minutes, sometimes it's 45, sometimes it's an hour and a half. So it's nice when you only have to do, the maximum amount once. And that's why I like this setup like this. But again, those are just things to think about. So I'm going to present some results where we compared our eDNA results for coastal cutthroat trout to electrofishing results. So first, I want you to look on the left side of the screen where you see the electrofishing results with blue bubbles. And so what you're seeing here is that the sample sites on the stream were in the middle. This is from a stream in Washington. And you're able to see, because there's blue bubbles at sites 1, 2, and 3, that there's a detection of coastal cutthroat trout with electrofishing. So yes, it was at all 3 of those sites. And you can see that sites 4 through 8 are gray, which means there were no detections of coastal cutthroat trout. The results for eDNA show detections at sites 1, 2, 3, and 4. And so you're able to see that with eDNA at this one site, just this example I've shown here, we have a detection with eDNA above where we detected for electrofishing. And you'll see that the orange is less dark. And so I presented the results here to bring up the conversation, because one of the big things people have to decide with eDNA, it's really obvious with electrofishing if the fish is there or not, because you see it, right? You catch it in your net and you see it there. With eDNA, you don't necessarily see it. You're trusting that the detection coming back with DNA is because it's actually there. And I'm going to show some things in our presentation about different times that you can get false positives or false negatives, and with eDNA that you have to think about. So what you're seeing here is essentially a detection with one of the 4 replicates that you saw. And so policymakers, managers have to make decisions. Most people decide that one detection in the replicate is considered there because the DNA is patchy in the environment. Every scoop of water that you take isn't necessarily going to have that species DNA in it just because it patchily comes down the stream, just because of how the stream water is being processed. But there are different groups that are considering, they want the majority of their replicates to have it before they would decide that it would be a positive or not. So again, looking at those results, what you're looking at here is a variety of streams. The stream names are down the center, and these results are presented in a mirror. And so site 1, for both of those streams are essentially near the center. So with electrofishing at all of these examples, electrofishing identified the upper extent of fish at site 3, but you're able to see with eDNA that we have detections above that and at varying strengths. So we have the bigger the bubble, the greater the strength was, the signal was in the lab, essentially saying there was more DNA in that sample. And then you're able to see on the color of orange how dark or light it is, how many detections that there were. And this one we had the 9 different replicates that we did. And so you're just able to get a sense of, this is what one replicate looks like. Is that a positive detection? This is what 2 to 3 looked like. Is that a positive detection? So I think those are considerations to think about as you're thinking about this work. The results of that study suggest extending the upper extent of fish beyond the known boundaries. So we were just using it to identify, distribution edges. It's one thing eDNA does really well, because all you need is a presence absence of if it was there. But you'll see it also isn't as simple as presence of absence, is it there? If you had taken one sample at all these sites, would that have been the one that came back positive? And we did an analysis where we actually did that. We took out, of our 9, would it be the ones that you would necessarily get it? It wouldn't be. So I always say it's worth taking more replicates than one, so that you increase your ability to get detections of something you're interested in. It's easier to take the samples in the field and decide not to process them later than it is later to, want to go back and do that. And so essentially this results are showing as you would be going from downstream to upstream. Electrofishing is on the right side, and it was the percentage of sites that had fish, using electrofishing. And so the upper extent of fish dropped out between sites 3 and 4 at most locations. We designed the study to actually be that way. We were trying to have it end between sites 3 and 4. But you'll see with eDNA that we get detections all the way up, so 250 meters past where we saw the uppermost fish with that approach. And so that suggests that fish likely move beyond that, especially at sites that have what I call soft edges. It's not the places with the big waterfalls, it's the places where they kind of putter out, season to season or year to year. We also found examples where eDNA may actually be detecting eggs in the ground. And so at this site, we had detections at all 8 of our locations in an absence, in locations with electrofishing. And so we speculate that it had to do with eggs being in the ground in this location. There's other work elsewhere that has actually shown eDNA can detect eggs in the ground, so you can get a sense of red locations and things like that using eDNA. And we actually also think that eDNA might amplify in a sample even when the fish is not actually there. So you'll notice that at sites 1 and 2 with electrofishing, we got detections. It's low, but we saw the trout there. We pick it up with eDNA. But then we also picked up this site, and it was like, what is going on there? So we went out to the stream to look at it so that we could try to understand more, and it was a game trail. And so the thought was maybe the signal of cutthroat trout, have them entering elsewhere, was being dragged across the stream. And with eDNA, we were picking up the signal. And so I bring that up because, they're in the system, but also probably not specifically at what was that site themselves. The signal was being dragged by other animals. And eDNA does really well at detecting trout in habitats that are hard to sample with electrofishing. So in and around large wood, if any of you have tried to sample in and around large wood, it can be really difficult. The fish are really so much better than us. And so it does really well, around large wood, and it also does really well in wetlands. As you're able to see here, we get better detection with eDNA when a wetland was located above the stream than when it wasn't. And so now I want to present some results with a technique that's broadly called eDNA metabarcoding. In our group, we do multi-gene eDNA metabarcoding, which is the same thing as metabarcoding, but just on more genes. It's like we do it twice or 3 times. In our case, we do it 48 times. But the point is just what you're doing with metabarcoding is different than what you're doing with that single species eDNA. So remember, single species eDNA, you develop the primer, so it'll only tell you coastal cutthroat trout and no other species. You make sure ahead of time that your only detection will be coastal cutthroat trout. Because when you get the signal back of a yes, that's what your answer is, yes or no. You want to make sure it's telling you the one species you're interested in. With eDNA metabarcoding, you actually look for a part of the barcode that will tell you multiple species. So you look for a place that's going to tell you Chinook, coho, coastal cutthroat, and rainbow trout all in one. And what you ask for in the end is you get all these sequences back. So you end up with A, C, G's, T's, and you then take the sequences and you map it to the species. So you have the barcode that'll tell you those 4, and then your data that comes back off the sequencer, well, then you have a data pipeline processing step that you map it to which, this one's coho, this one's Chinook, that one was cutthroat, no micas here, or something like that with your results. So That's why what the example you see here is you essentially see two different fishes, and you see us targeting, these are different gene names. So the ND2COI12S is considered a taxon general metabarcode. It's often the one, there's a barcode that everybody uses for ray fin bony fishes, and it comes from the 12S gene. And it does really well at generally telling you what fishes are in your watershed if they're a ray fin bony fish. And it's used globally. And then for salmonids, some people also use ND2 and COI for the different subset of those groups. That's why I pulled those out as an example. And again, then you get the DNA and you're mapping it to the species at the end. So in this step, it's different than the previous one where you're just going to get a glow of fluorescent green and it's going to tell you yes or no, was your target species there. With this one, you're going to get the output of all the sequences and you have to do, you know, like a bioinformatics step. And so you have to have somebody with the skills to code to be able to then sort those sequences into what species is of interest. The cool part about this approach, and this is the part where research right now is in, and I would say if you have really advanced, somebody with really advanced skills, you could be doing it now in monitoring, but this is where we're going. I would say the single species approach right now is set up to monitor, and there are a couple projects out there right now. There's a couple genetics platforms that actually allow you to do multi-parallel qPCR, so you can do up to 10 species at the time. So essentially you're just doing each 10 single species analysis in one sample. And that seems to be working well for a lot of different groups. And it's easy to interpret when you get the data back, because it's just like for each species, yes or no, essentially is, you know, what you get. Now, again, when you get this back, you're getting that. But when you analyze the data, oh yeah, then you get this. And you can say, you know, okay, you know, 24% of our sample had omicus in it, 9% of the stream had cutthroat trout in it, you know, 6% were Chinook. And so you get a better understanding. You got coho, we have that many sculpins, and you have lamprey, and then you can tell, it'll tell you everything else that you looked for. So you have essentially a better understanding of a variety of species that would be in your sample. And it just gives you so much more information. So using that approach of the multi-gene eDNA metabarcode, we looked at Fall Creek that flows into the Alsea watershed in coastal Oregon. And this was our electrofishing results. So we have the young of the year trout, which in the field with electrofishing, you can't distinguish cutthroat from rainbow. So we found them at all the 4 different sites we sampled. We have cutthroat, we have rainbow at 3. You know, this, so I should say this is the downstream locations and it's moving upstream in the same stream, so in Fall Creek. And so it makes sense, you know, the species, you know, are in our 4th order streams. You're seeing the Chinook juveniles, coho juveniles, you're seeing your sculpin, some of our dace, the lamprey, some of the different amphibians, and our native crayfish. With our eDNA results, between the 2 approaches, we had 87% accuracy. And so we're seeing some detections with the eDNA approach that we don't see with our approach. So for example, eDNA suggests that rainbow trout are at the most upstream site. And so you're able to see that here where electrofishing said no, eDNA says yes, but that makes sense because young of the year, we can't distinguish what it actually is, but eDNA genetically can distinguish the 2. And so you're able to say, you know, yes, at that site, rainbow trout were there. eDNA also suggests more lineages of sculpins, and that potentially they're a cryptic species complex. And so this is how many sculpins were identified with the first approach. We had at this site, we had Doug Markle, who specialized in sculpins in Oregon, who used to lead the Ecology lab ID our sculpins, and said, yep, those are the 3 you guys have in that system. And when we came back with eDNA, we had this 4th category. I just called it the sculpin spa category because it ended up having 4 to 14 more lineages that I just categorized into the 1. And so again, just to highlight that rainbow trout piece, you're able to get detections with eDNA over the electrofishing, and I just wanted to show the results for the different genes we use. One of the benefits of this approach, if you do multiple genes, you don't have to, you can just run one gene and look for your results. But as you have built-in redundancy, and we liked that, especially as we were working with managers that said, I really want to be sure that these fish that I'm interested in is there. And so we said, okay, well, we'll look for it on 3 different genes, and we'll look for it in 3 or 4 different replicates. And then if it's there, and we get detections across multiples of those, it increases the likelihood that it actually is there. So this is just to show you, this is the eDNA data summarized all together. And then that's to show you the 3 different genes and the different taxa that it targeted. So if you notice that the bars of the checkerboards aren't present for one of the taxa, it's because we didn't use that gene to develop them. So for example, we use COI for sculpins and for the salmonids, but we don't use ND2 for sculpins because it doesn't work as well. So that's why over there you see the different groups, represented with the checkerboards. So kind of coming back to that idea of sculpins and the different lineages, this example is from the Trask watershed that's part of the Tillamook Basin in west coastal Oregon. Again, the electrofishing data is on the left, the eDNA data is on the right. With young of the year with eDNA, since you can identify the species, it's grayed out there. Because with electrofishing, we were targeting vertebrates, the crayfish, the mountain beaver, and BD, the fungus wasn't detected because we weren't sampling for those with electrofishing, so they're grayed out there. And you're able to see just the kind of detections across the different species. But specifically, if you focus in on the sculpins, you can see that in the field, and we tried to do a really good job knowing that we were going to get these results, that we were actually identifying them well. We thought for sure the saddleback torrent is at that site and only that site. It turns out it wasn't at any of them based on the genetic information. So what we think is a torrent is actually probably something else genetically. We thought we had reticulate sculpins at all 4 sites. The genetic result says, great, you have it at 2, not 4. So there's some sort of cryptic species that you're calling reticulate that's likely something else. And maybe at the one site, it could be any combination of these. This is how, again, remember how I said it maps? You get the sequences back and then it's mapping to the known species. It's mapping to these. Notice how I call it prickly-like sculpin and margin-like sculpin, because other systems where we would call prickly and margined aren't those fishes. But there must be something because genetically it's mapping back. So there must be some sort of prickly complex of sculpins, or what we're calling certain sculpins aren't actually what they are, which comes back to if you go with this approach where you go with the metabarcoding, there's a really important piece to that, which is that you have a genetic library of information. Because this sits on when you have to map it back, you need to have the genes connected to the specimens. So one thing right now that they're working at the Oregon State University fish collection is to actually have vouchered specimens that are going to be linked to sequences for the state of Oregon. ODFW has partnered with OSU. There's a PhD student named Emily Dezijek who's working on the project, and she's trying to map 3 species of every single fish species in the state of Oregon, so that essentially we would have this database that when studies come out of anywhere in the state, we'd be able to say something about what it's mapping to. And that'll be really important, especially for a lot of these sculpins and things that are a little more cryptic. It'll help us get a better sense of what we're actually finding. So again, this is the same study in coastal Oregon in the Trask watershed. And you're seeing that eDNA detects sparsely distributed species. So just as an example, I mentioned that it can detect mammals. We had targeted beaver, which included mountain beaver because our metabarcode could detect them. We didn't actually detect any beaver, but we did get mountain beaver. And we were able to detect BD. I just bring that up because I think an important thing to note is, putting somebody out in the field to collect a sample and then you bring it back. There are many species and things you can look for after the fact, or multiple groups could come together to fund projects, and you could fund it for plants and water quality parameters and pathogens. You can look for human pathogens in addition to the ones that are for animals, and you can do a variety of things that way. So this example is from Big Water in the Lake of Oregon, and so the sites are noted down the side. So we have reservoirs, large rivers, and lakes. And what you're looking at here is the species down the right side. So we have ringed crayfish, invasive crayfish, we have New Zealand mud snail, and then we have 3 different plants up top. And so essentially we were looking for, can you actually pull from the same sample a plant and an animal? And the answer is yes, you can. You can do that from the same. We weren't sure if the plant signal was going to swamp the animal signal because they're so much more plentiful. And so we're working on prepping that, but this is over time. And so you're seeing the different spikes and what it is. And this comes back to what animal are you focusing on? And are you sampling at the right time to get the best detections from that animal? I mean, just think of salmon coming back to spawn, or amphibians that are, maybe move into the streams at a certain time of the year to breed. Others, might be stream obligate, but are you sampling at the right time for what it is you're focusing for? And sometimes it helps to do like a pilot sample to make sure you're, are sampling at the right time period for the species you're interested in. And for the big water, we sampled less volume of water because we can't push as much volume of water through our big water environments. So that's just another thing to think about. We were able to do 750 milliliters, but with less volume of water, you have the less ability to detect. And so those are kind of like the trade-offs you're trying to figure out what you need. And so as part of the pilot study you might do in advance, you might say, what's the best volume of water, the highest amount of water I can get at all the times I'm going to be sampling? And you might have to test a couple different levels before you decide, okay, we're going with 3 liters, or we're going with 1 liter, or whatever it might be based on what you're focusing on. And one last study I'm going to present, and then I have a couple wrap-up slides and we'll be ready for questions, is just to show that with the example of the eDNA metabarcoding, where you get the sequences back, you can also say something about the genetic variability of the species. So this example is of coastal cutthroat trout. As you can tell, they're my favorite fish because I present all the time on them. But this shows essentially the different streams we sampled. So the sampling sites are shown where you either see the pie or if it's empty. We sampled there, but we didn't get a detection of coastal cutthroat trout at the time we sampled. It was summer sampling. And so you're able to see the genetic variability because we're showing the different haplotypes here in the pie charts. So the more colors was the more variable it was at that site. And so you see the high genetic diversity in our coastal rivers. You see that some rivers have private haplotypes, which essentially just means it has a unique haplotype only found at that one site. So one of our coastal rivers had that, as well as the Umpqua in the places that we sampled. And that there's moderate diversity in the Willamette and Umpqua rivers, but those two don't actually share any diversity with each other for coastal cutthroat trout. And that there's lowest diversity in the Rogue River, but they shared sequences with the coastal streams and the Willamette watershed, suggesting that there's some mixing between the species and those different river systems. And so this is just to show what you can do with data that you would get back, if you went the route of eDNA metabarcoding, that you can get at pieces of genetic diversity. So again, in summary, eDNA allows you to get at different aspects of biodiversity. It's more sensitive than our traditional approaches. You can get at distribution, so you can say something about contractions and extensions of species. If you do the metabarcoding approach, you can talk about the cryptic species complexes, like you saw with the example for scopins. I think we're going to see something like that also with our suckers and daces, as I'm seeing some of my other information coming out from the Willamette. It does really well essentially when species are in low density. So when you have, 3 or 5 or just a few, you're able to detect, with eDNA. You can detect emerging pathogens such as BD, BSAL, whatever that might be, whatever you might be interested in. And again, it does well at detecting early detection of invasive species. And so you can tell, when crayfish might move into a stream or tributary you're interested in. And so as you think about all the different things I talk about related to eDNA, there's different considerations you have to make essentially as you go down, this list of things to think about. Are you studying in a lake, river, stream, pond, wetland? Because that's going to determine the amount of volume of water you sample and how many locations you might want to sample. How big is it? Can you just be, think of a giant reservoir like Crane Prairie. Our results came back, we went to 4 sites, each of them got different results. So essentially it was detecting different things. It seems to be detecting things that are more localized. We were sampling during seasonal low flow in summer, and the eDNA literature suggests that in spring and winter, when flows are up, in your streams and rivers and even elsewhere, that the signal travels further. So those are things you want to be thinking about in the environments you're sampling in, for the species you're detecting. For the different species you're looking for, when are they in the system? So is your timing for when you might sample correct? What are the objectives of your project? Again, do you want to get at, the presence, absence, the relative abundance? Is there any variability in your ability to detect signal? Essentially what's out there, higher water temperatures or air temperatures. Just high temperature degrades your DNA, higher pH degrades your DNA. How you're transporting your samples, those are things you have to be thinking about. And then all the decisions you have to make have to do with pore size of the filter, the volume of water, how many replicates you're taking. Are you processing field blanks? The reason to do field blanks is it tells you something about your contamination. And it also allows you to be able to say, we detected that really rare species at that location, and they'll say, well, you just dragged it there on your waders. And then you can say, no, we didn't, because we took a field blank, and that shows we didn't drag the signal, it came from the environment. So the benefit of field blanks, it kind of depends on what your objectives of the project are. Is it really important? Because the data that you provide to whoever you're giving it to needs to be kind of scrutinized at that high, robust level. If so, you want field blanks. Again, do you want multiple genes to detect what you're doing? You can do multiple genes in both the single species approach as well as the metabarcode approach. Is that something to think about? How many sites are you going to? And again, the timing of sampling. So I feel like there's so much information there, and different things to think about with eDNA, but it is, where the field is going, and it's kind of the cutting edge of where we're at with stream work and biodiversity assessments. So with that, I'll take any questions. So your first question had to do with how far signal travels within streams, rivers, lakes, ponds. So signal travels the furthest in rivers, oceans, essentially water that's moving, and water that's moving at high flows. So if you think of a stream first, stream signals traveling less quickly, just because it generally flows less than a river. That's not always the case, but that's the general idea. Ponds, it tends to be still water, wetlands still, it's not moving at all. It's residual in there for a while. So streams, the signal tends to stay up to 90 days. Sometimes it's gone as quickly as 2 to 3 days. Summer, so think of our seasonal low flow. Detection, or the signal's not traveling more than 50 meters. So the sites that I showed when you saw all those orange dots on this side, each site was 50 meters apart. And you could see that there were holes sometimes in the middle. So the signal was not always, arriving downstream. But there's work that shows that, during spring and even the sites in that study that we sampled in March, because we were trying to sample within the window for the Forest Practices Act. And so we were sampling March to June. The ones in March that had higher flows, you could tell the signal was traveling a little bit further. I'm not exactly sure if it was a few hundred meters, but like the signal seemed to travel further. And that's backed up by other studies in the literature that essentially show during winter flows, sometimes it's up to 1 kilometer your signal might travel. And then the same thing, it's in rivers. So a lot of the studies have been done in European rivers, looking at distance. And so it's been 5 kilometers I've seen, that signal's traveling in the rivers. And I should also say a lot of the literature related to the metabarcoding is coming out of Europe. They're really big on just full biodiversity assessments in their studies. And a lot of the work on single species work is coming out of North America, and we sort of push, the one species and over there. So like both approaches are being developed and used in a variety of ways. So to answer your first questions, it depends on the environment you're in or the water body type you're in. And it depends on the season that you're in. But I would say if you're thinking of streams during seasonal low flow, it's probably less than 50 meters.
You mentioned how it might be possible to get a false positive, with dragging just an animal walking through stream. But what about false negative? I noticed on one of the slides with the electrofishing technique versus the eDNA technique, but I think the Pacific giant salamander was shown in all 4, but in only 3 in the eDNA. How would that happen?
So right now with the metabarcoding eDNA that we do, like amphibians are our nemesis. We cannot figure out what's happening to them. So one thing in that, when I explained at the beginning how you have your sample and you go through the 6-step process in the lab to extract the DNA, they're in the water, there's what's called inhibitors that essentially, it's kind of like they block your ability to identify the DNAs of there. So there's natural tannins and thing in the environment that actually acts as inhibitors. So you have to remove the inhibitors. Sometimes in the removal of the inhibitors, you remove some DNA. And right now we're trying to figure out what's going on with amphibians for us and why we're not always detecting them. Or the DNA of like amphibians is like particularly yummy to something else in there that's eating it. We don't have the answer right now, but the whole amphibian group for us are causing us some issues that I think might have to do with inhibitors, or I like to think of yummy DNA, but I don't think that's actually probably as likely.
Kind of similar to Aubrey's question, you had talked about the strength of a signal. So my understanding with eDNA was it was that Cousins-Jackson question, not density. Is there a way to interpret density with that?
Yeah, definitely. A lot of people don't want to present the results that way, but there have been really strong correlations between traditional approaches and eDNA showing you can get at relative density, and you can actually say generally is the species rare, or was that population in your system rare? Is it more common, or is it super abundant? So I would say you can get a general sense of the relative abundance, but you don't have necessarily the sense of was it 10 of them detected, but you can have just a general sense of what's there.
And can you do that with both approaches or just with that?
You can do that with both approaches. And there have been a variety of studies that have been published in the literature, like tens to hundreds of them now, that have shown that there's correlation between traditional studies and that. And so essentially systems that have bigger fishes, so think of like the Willamette River, that just have bigger body fishes, it correlates better with biomass. And streams that are smaller correlates better than with density. So essentially you're just counting the numbers versus, how many box sizes in there. And it makes sense because you're getting back DNA, from the different sized animals. The other thing with DNA that can really mess up your ability to interpret abundance is when an animal is dead, it decays and its DNA is just blown out of the system. So you think of, going for spawning salmon, especially in our systems, and you can see the signal just spikes. It goes from like tens to thousands, when they spawn. And so because when they do die, their body just lets go of all their DNA. And so you'll get a huge spike in the system. So there are certain things that might not be exactly tracking with abundance, like the dying out of certain species.
Diversity with the map with the different systems. In the Willamette, I noticed lots of Cascade Range rivers and no Coast Range side rivers in the Willamette. So what do you got against Coast Range?
Well,
the Marys and Luckiamute.
I know. I have nothing against it other than my crews that went out were going to federal land. So they were going to the BLM or Forest Service land for that project. And so they were going to sites they were already going to already. And so that's the reason for it. No bias against you. And actually, I should say, I have a new project that's starting that the Luckiamute will likely be a part of, where we're looking at agricultural channels. And so Guillermo Giannico and Ivana Arismendi are doing like traditional sampling right now of minnow trapping in a variety of the agricultural channels. And so they brought me in to do some eDNA sampling that'll go alongside their minnow trapping, and then it'll be more extensive. We're going to add an additional like 45 sites. And I know that right now we're figuring out what sites, and some of them are in the Luckiamute watershed. So yeah, there's that. And then we have, over the 21, 22, and 23, I did boat and backpack electrofishing on the main stem Willamette, along with eDNA sampling. And I took eDNA samples of every tributary contributing to the Willamette River, so essentially just above the confluence where it met with it, to have a sense of how many species are contributing to what biodiversity I see downriver. And we're processing all those samples now because we wanted to get them all in and then process them all at the same time in the lab. So we're just processing the eDNA samples now. I have the traditional results back that I'm presenting at the TAP Talk tomorrow, but there'll be some results from the Luckiamute in that as well. Yeah.
So for more traditional water quality monitoring that we do in association with DEQ, they, in order for that to be made publicly available, they require us to develop a sample analysis with them in order to make sure that the data is robust enough for them to be comfortable making it public. Are you aware of any kind of analogous standards and practices that we could use if we wanted to pursue eDNA?
So there's a paper that's Goldberg et al. 2016, and it lays out eDNA guidelines. And essentially the group of scientists came together to put that publication out, and they tried to come up with a set standard, but they couldn't agree. So essentially they have guidelines for all of us that touch on the different things, I talked about, but you can use that as you start to think about the different pieces of the work. And Karen Goldberg's out of Washington State, so she's rather regionally local.
Okay,
So-
they'll go to the Zoom folks if they have questions.
Have you heard of the private forest board?
Yes.
So do you think this would be a useful tool? The way they made the new maps was based upon modeling. I don't know any of those details. But on our property, there's a stream that I would be shocked if there were fish in it. That's now a fish-bearing stream, and therefore that changes the buffers. And I'm sure there's lots of other landowners that have the same issues. So, right now their plan is to invite one of the foresters out with a representative from ODF&W, and they look around and decide whether or not they think it's correct or not. So it seems to me like this would be the perfect tool to really get the right answers, or excuse me, to be accurate in these maps. And so the big issue would be cost. So what would be the model that they use?
Yeah.
That cost versus actually going out and getting samples on these ones that to be a tiebreaker.
So we, written into the current Oregon Private Forest Accord, you can use eDNA to determine the upper extent of fish. Now, it was added in as an accepted tool. So it is a tool that would be considered in that context. It wasn't used to inform, there's two models that are being considered right now under the Oregon Private Forest Accord, the Franson model, and then one that was developed for Western Oregon that's across land ownerships. The Franson model is actually developed on Weyerhaeuser land in Southwest Washington. I think that's the model you're referring to because it's the first ones that have been pushed out by the Oregon Private Forest Accord. And it's both of those approaches are based on electrofishing because there's less controversy for people to agree to use that as the tool. So I agree with you that eDNA might be a better tool to use. It's more sensitive, and a variety of studies across, continents have shown it's a better tool. But I think to get agreement by people that were part of the decision making for that policy, they went with electrofishing to inform the model. But I think it would be, I think if we used eDNA, it would push fish further up. And
So I don't think that's an issue though. It's all or nothing. It's either the fish-bearing stream, the stream is either a fish-bearing stream or it's not a fish-bearing stream. The stream is either salmon, steelhead, bull trout, or just some other type of fish. So it doesn't really affect that.
my understanding is that streams would drop out where fish were. It could be in the middle of like a main stem tributary. If fish dropped out in the middle, it would drop out in the middle. And so that would be the end inflection point of that distribution edge. And then everything above that point would be no fish, and below it would be fish. And that's my understanding.
Okay, because right now, the way the maps are, just
It runs it up to the end.
It lights it up to a certain point, and then stops. And so I know in our places where there's a county culvert,
And I think,
uh,
models can only do too well. You need good data to inform the models, and the models can get better. So I think, it's important that we get better data from the field to inform those models, whatever tool would be used. I currently have a postdoc that's actually trying to create the model that will be across the west side of Oregon, Washington, Northern California for the Oregon Private Forest Accord. And so we're working on, sampling in a variety of watersheds to essentially do just that, to get better information to inform the model that would be used, because there are limitations and basins that weren't initially sampled, and then, it's being applied for all of them. I mean, I think it's bad for the west side. The same model is being applied on the east side. They don't even have coastal cutthroat trout, and it was developed for it. But we don't have anything over there unless it was applied. So right now, it's just the best information that there is. But, our plan too is then to develop one for each of the different watersheds on eastern Oregon, so it'll be appropriate to the fishes in the basins there.